Okay.
So guys, we'll start just by rolling through vocab here and see how you're doing on memorizing vocab. Does that sound reasonable?
Yeah.
And then we'll ask specific questions and we can draw or whatever. So we have, these are all examples of what?
Macromolecules.
Wait a second, I didn't do that right?
You click
Yeah.
it.
Click, flip. Yay, good job. Have some more candy.
There's not a lot of us. Should we?
Have more candy? To make yourself sick. My policy is you vomit, you clean it up.
Okay.
Which I don't have very often anymore since we don't dye fish sharks anymore. Any other questions? You have to wait? Yeah, well. Chicken strips. That's the biggest seller, huh, the chicken strips? Elaine's mom. My daughter ate like that much candy in 2 days. Trick-or-treating. I went to look at the candy bag and in my book, what happened to all this candy? Did your dad eat this? And she's like, no, I ate it. No wonder she hasn't been feeling well. And she was crabby yesterday. Hydrophobic, water hating, hydrophilic. Wait, hydrophobic. Substances do not dissolve in water. What does this card say? What?
Maybe we should start with the other side first.
Wait a sec, there's the audio component. Does that mean I don't play this? Oh, I hate those voices.
Oh yeah.
What is this? It's not really a card.
No. Here. Go to options.
Maybe you should come to this. Where's options? Down here. Got it. But we'll see. Lower left and say.
And then
We don't want up.
wait.
Try advanced.
Yeah.
No, that's just
Oh
audio.
no, that's audio.
know.
There's like one thing that you can do where it's like you flip the place, so it like shows you like the front of it first. Because right now we're just looking at the back of the flashlight.
Well, the first one did that. How about play? Duh. Oh, I'm turning that voice off. That's awful.
Yeah.
Oh, I clicked on that and it went.
I think you just actually click on the
Wait, I'm listening, Connor. Hold on. Okay, flashcards, Connor, advise.
You just like click on structural polysaccharide.
See, this says click to flip. Okay, structural polysaccharides of plant cell walls are what? Cellulose. Very good. Now can I go to the next one?
Yeah.
No, I think that's that one.
Enzymes.
What? I don't get why there's not a.
Just click it. Oh.
None of us would have guessed that.
Yeah, that don't work as efficiently.
Right. Today in lab, we had a group that used water that was 50 degrees for the yeast solution and it had 0 bubbles. Like, oh, it didn't work. I'm like, no, it did work. It's great. But it was boring. No bubbles. So enzymes, guys, tell me about enzymes. What do you know about them? How they're designed?
They're catalysts.
Catalysts.
Speed up the process.
They speed up the process.
They're designed for one type of chemical.
They're designed for one kind of chemical. What do we call that chemical? What do we call that one substance? Substrate. So they're designed for one substrate. Anything else you know?
The pH temperature and inhibitors like change, like can affect the.
All right, so they can produce products faster or slower depending on environmental conditions. I'm trying to shut her up. I already shut her up once. Stop it. I don't want to hear you. Okay, so here's another one. They're made of carbon, hydrogen, oxygen. Hi. We're working on some flashcards, which I'm not doing a very good job at here.
What's the difference between
Carbon, hydrogen, oxygen. What's the difference between carbohydrates and lipids? Is that what you said, Daniel?
Yeah.
Good job. Next card.
I would say structural.
There is an amino acid which you need to know the structure of.
What's an amino acid? The part box.
Am I looking at the wrong side of the card?
Yeah,
Yeah.
we're looking at the back side.
How do I look at the right side?
There's usually something that says like term first or definition first, but I don't see it.
Flashcards, list, okay. So, original, alphabetical, take into account. I obviously don't spend time on this site. Alright, well,
It's kind of like playing Jeopardy.
it is. Okay, amino acid, structure,
What about the bottom part? Can you
What's
see
that
it? It's like
bottom
on the
right
bottom.
thing?
the bottom part,
Bottom right. No,
Bottom
on
to
the,
right.
On the thing.
that,
Yeah. Do English or chemistry? Click on. Wait, can you click on that?
chemistry or English?
Click English.
It's not a click, you press the
Or press
key
the
on the keyboard.
Oh, no.
Press a 1 or 2.
Okay.
Did,
So
but.
then you have to go out of that and then press like the other two.
Protein. What do you know?
Made up of amino acids.
Amino acids, keep going.
They're chains that are on the outside of the cell membrane.
Ribosomes make them.
They're found in the cell membrane, made by ribosomes. Anything else? What are the polymers called? I heard you talking about monomers and polymers earlier, Nicole. What's protein's polymer?
Oh, I remember learning that.
Poly what?
Oh, something with P.
Polypeptides.
Peptides.
Polypeptides. Anything else we know? What kinds of examples did you have in your notes of proteins?
Like foods?
Hair.
We gave some specific functions, some examples or functions. What? We said they're found in cell membranes, so some of them transport. Logically, what else do proteins do? Logically.
They make up muscle.
They make up
They fight
muscles,
disease.
they make up cells, they fight disease. And enzymes are a great example of proteins. All right. Let's go through these kind of quickly and then we'll see what questions you have. What does this thing look like?
Oh, it looks like the thing that we made.
It
Polysaccharide.
does, it's a polysaccharide, so you guys will have to recognize all the pictures. Hopefully everyone got that, I put those in your notes. So make sure you know the pictures for each of the macromolecules. You'll just have to identify them by sight, I think that's easy, now that you've looked at them for a long time. And we would guess that this is a polysaccharide because it's got the carbon ring. Here's some examples of polysaccharides. We didn't talk about chitin. It's found in bugs, exoskeletons. What would you do to break these bonds?
Add water. Hydrate.
Add water, and what's that reaction called?
Isn't it also condensation, or is that?
That's the other one.
Yeah, that's
Putting them together.
dehydration.
Yep. Carbohydrate structures. What? Because they're all hydrophobic.
Oh, is that the back of the card?
That looks like
What?
That looks like the back.
Is that the back of the card?
I don't know. I can't tell.
And then that's the
Oh,
back.
click 1 or 2, or click 1
Where's
on your
mine?
keyboard.
Wouldn't that be it? Yeah, that was the back. Yeah.
What's hydrophobic?
Afraid of water.
Afraid of water, fats, phospholipids, steroids. They're hydrophobic. They don't dissolve in water, right?
No.
So they're hydrophobic. Carbohydrate molecule made of 2 monosaccharides. So easy.
What's a disaccharide?
What's a disaccharide?
You
You like the pictures okay? You're okay with those pictures being saccharides, disaccharides? You don't have to memorize that it's glucose or galactose. Well, maybe glucose, because we built one. But you don't have to know the names of individual sugars. Sucrose is made up of 2 monosaccharides bonded together.
Oh, that's the back of a card.
Help me, I can't view
Press
next
1 on your
I
keyboard.
did.
There we go.
Why would table sugar be considered a
It's
disaccharide?
made up of two monosaccharides.
Made of 2 monosaccharides. Now if I click next, is it going to stay on the other side?
No, you have to click it really fast, the one, so we don't see the back side.
I click 1 fast and
You
then
click,
advance.
no, you click the advance and then you click 1. Yeah, it's good.
No.
Oh, that's
you
your
know
You don't know that one.
Yeah.
What makes up amino acids?
Um,
Amino acids are the building blocks.
Is that the back?
This is a harder one. Carboxyl group. Remember that? The R group, the hydrogen, and the amine group.
oh, yeah.
There we go. So you guys actually have to know the names of the parts.
Wait, what are they? The R group?
This is the carboxyl group over here. The R group is where it attaches to another molecule. And we're going to talk more about this when we get to protein synthesis. And this is the amine group. That's in your notes. Amine, carboxyl. That looks kind of carboxyly. The R is where it attaches to another molecule.
Is it a molecule or is it?
No, it's just a location. Yeah. So there's a bonding point right there. It bonds onto some other molecules to give it specific function. Okay, now do I have to hit the 1 again?
Yeah. Good. Does not dissolve in water.
No.
Oh wait, it does
It
dissolve in
does.
water.
You
Philic.
tricked me. I flipped it and it. Okay, hydrophilic is water loving.
Water loving.
Hey guys, remember we're not doing the plasma membrane until next test, right? It's just 2.3 and just 2.4. So this test, all you would need to know is that this region is hydrophobic, fats, oils, and those
Wait, so we
groups.
don't have to draw it yet?
No, just 2.3 and 2.4. Let's see. What are these examples of? Where are they found?
Nucleic acids.
Where are they found? In the nucleus. What is their building block called?
Nucleic acid.
What's their monomer? Never mind.
Well, they have,
Nucleic
like,
acid. I'm trying to think of another example.
What sugars are they made of?
No, they have,
Yes. What are the monomers? I heard it. Nucleotides. So the building blocks. Thank you. I can't see Connor back there. So 2 examples of nucleic acids. And you don't need to know anything about DNA or RNA except that they're nucleic acids.
Nucleotides are the
Building blocks.
building blocks.
Nucleotides are the building blocks of
So it's like the little, like,
DNA and RNA.
nucleic
The
acids.
little things that like make them up.
Yep. Nucleic acids. Polymers, general term.
More than one monomer.
More than one.
Oh, it's actually more
large.
than 2.
So I think the smallest functioning protein's 40 and the largest
No,
is
it's more than one.
1,000.
it's more than one.
I was thinking of a disaccharide.
Yeah.
There's no
I
di.
won't be that picky. Oh, look, it flipped back.
Trisaccharide.
Hemoglobin is such a fun word.
I know.
Hemoglobin. I'm not
Protein.
flipping. Let's see, you guys had hormones. I think I that right in your notes. Antibodies, proteins, hydrolysis,
That's like.
combining or taking apart.
Breaking apart.
Breaking apart. Hydrolysis requires water to break down the molecules. What do you know about lipids? What are their basic units composed of? We looked at 2 different ones. One was the phospholipid. The one we looked at in this chapter was called the... It had 3 little tails instead of 2.
Oh, triglyceride?
Triglyceride. The 3 little tails are called what?
Glycerides.
They're chains.
They're chains of
Hydrogen.
fatty acids. And then the other part is the glycerol molecule, right? Let's see what they said. Yeah. So you would identify this easily as a lipid, right? Here is the glycerol component, and then it's got 3 chains
Fatty
of fatty acids.
So
That's what red blood cells.
a triglycerol
Whiteboard.
is a lipid.
Because it's cute,
So
and
cute.
that's my favorite shape of cell because it reminds me of donuts. So,
Solid.
Solid.
Good point.
enzymes we went over. Specialized proteins that help the body's defending itself against foreign invaders. Do we know this? Antibodies, but we didn't put that in the notes. So what group are antibodies in for macromolecules? Proteins. Why are these things different if they're made up of the same building blocks?
Because
Some are
they're
in
more
plants.
complex.
What?
They can be more complex.
Some are more complex.
Different
They're
functions.
made up
They
of
have different functions. Which of these is found solely in animals, like birds, humans, cats? Which one? Glycogen.
Isn't it made up of like approximately 30,000?
Monomers? I don't know. It might be.
It was
Oh.
3,000 or 30,000, but you know, they're basically the
For
same.
glycogen,
Yeah.
where the glucose molecules are arranged, we didn't really study that. We already did that. That's made up of nucleotides. What are carbohydrates, proteins? Are we at the beginning here? What do these things have in common?
They're the different macromolecules.
And what
40
makes a
types.
macromolecule? The one atom that we see in all of them, carbon. Hydrophobic, we know. I think we did these already, right?
Yeah, we've already done these.
Okay, so let's see what other questions you have, stuff you're not as familiar with. I think you guys, once you get the vocab down, this kind of intimidating when you start off. If you get the monomer, polymer, saccharides, sugar, lipids are fatty acids. You start to get all that vocab down, and you're pretty good. I think. So, what questions do you have for me? What? What's my favorite sandwich outside? It's pretty good. What else should we go over? Let me look at my notes if you guys don't have any questions. Yes, Joe?
Do the format of the test, is it going to be similar to our last one?
Yes.
So are there short answers?
Yes. And you don't get hints on the short answers, although I'm sure you'll be able to guess. If you do your reading and you're studying, the short answers should be fine. You should be able to interpret those pretty easily. Just a reminder, make sure you know the little diagrams. And another examples, put those on note cards. So I know you hate note cards, but do you think they're helpful? You guys are afraid to answer me because you're like, she'll make us do it again. Do you hate note cards, Connor?
I would use it as a method of study.
Yeah.
Yeah, I'd read the book.
Okay. Any questions? Are we good? Yeah. You'll be fine. And we still have your note packet's always like the basis of material, but you gotta go back and look over your Cornell notes if you do a good job, and your book if you don't do a good job on Cornell notes. So skim over the reading, because it puts everything into context, so this is out of context.
So if we go over the notes, our notes, those
Should be
are
good.
more or less, and read through the book.
You can read through the book or look at your Cornell notes, either one. You're doing a good job on your Cornell notes. That's what they're designed for, is to study.
Thank you.